So we now have a brief discussion between all of the members of the panel, just talking about lessons learned from this session and how we might apply them to future studies across Asia. And my question, first of all, to HCA and to Isham is really about the importance of studying the pathogen or the microbiota at the same time that we study the host immune system. It seems to be a really critical gap in a lot of studies.
Sorry, I was mute. Thanks, Christine. Yeah, I think it is very important because now we know that microbiome is not just associated with one or two, but a myriad of functions, like be it normal functioning or a disease. So doing something, building something on top of that will help us in a lot of study, not just like one particular study. So if we do that in HCA, it will be very helpful. The data will be very helpful for all sorts of studies, like ranging from cancer to basic biology to infectious diseases, anything. And the other important thing is that here we have an opportunity to basically start and make a pipeline through which immune system and microbiome both can be captured together, which has not been done before. So setting up this pipeline is very important, and it's high time now that we start like studying them in conjugation, not just in some mouse models or germ-free mice and stuff like that. So I think we always get samples like these, and the important thing is to start looking at both the aspects, not just the immune aspect of it. So, yeah, I think.
Thank you. Ishan, you study specifically the pathogen in your atlas, and almost coincidentally got some contaminating human cells. I'm interested in knowing how we can do this combined dual species genomics more effectively. Ishan, you're on mute, I'm sorry. I've just asked you a question about how we might just move forward with dual species sequencing a little bit more effectively if we wanted to include the pathogen and the host immune response at the same time.
What was that mean?
Yes, please.
Okay, sorry, I didn't hear for a second. So yeah, I couldn't comment for other diseases, but I think that would be the way to go. So so obviously for dengue, sorry, am I supposed to be speaking or?
Yes, please go ahead.
Okay, sorry, I just doesn't match the screen. So yeah, I mean, at least for dengue, we can see in some studies already that apart from just sequencing the what's going on with the immunes, for dengue we can I think there was a study that maybe Ananya and Waradon can add to this as well, that it's now possible to sequence the host immunities and at the same time as the RNA of the viruses. So I think it's still quite at the early on stage. But in the future, I believe that would be the way to go. But as you mentioned, that in terms of resolutions, of course, you're gonna get like a lot more host sequences than the microbes or viruses. So yeah, so that technically that would be something that we have to work on as well. But I think you spot on that in addition to just look at either the pathogens or the host, as Ashita mentions, that these are sort of like into one basket, that we have to study them together. The question is that how would we improve the resolution and technically to not just to analyze them together, but to combine and build a model how they interact together. Yeah.
That multimodality is going to be a real challenge, and you can go ahead.
So I think the challenge is the fact that the single cell technology currently works on super Poisson loading, which essentially makes sure that you have the gel bead coming up with a the single cell. That's how it makes sure that a single cell is falling. But when you are talking about two different species, you have different size differences, and that super Poisson loading might not work as efficiently. So there are new platforms which are coming up, I guess. Dana Pair might be working on some people in our lab where they are trying to combine two droplets. So the idea there would be that if you have already captured a single cell of one kind and you want to capture basically non-viral pathogens, then you have to work with those technologies which have two different streams of cells captured, and you can combine them. And then so yeah, I think it's a technological challenge. Otherwise, to control the number of samples and the number of pathogens per the host, that could be a challenge. But like, yeah, at least for viruses, a lot of studies have come out already trying to look at this.
Very much. I mean, Faridun, in
So can I add a little bit further on, like what Varadom said? And this is because, we have TCR and BCR information based on like the 10x, right? But a lot of time we're struggling with, like what they are, like what they are recognizing, right? And then, like this is relevant to, like specific infection that we're interested in, and at the same time with microbiota, right? Like, can we know like what these T cells are recognizing, right? And now, like as Varadom mentioned, like there's some advances, especially in the context of COVID-19. Like there can be a dexamer attached to, like a specific epitope for, at least in the context of T cell, with particular HLA haplotype, and then you have a pool of potential epitopes for like SARS coronavirus too. But then like, can we extend more, right? Like, can we do more on like other like microbiome, and then like other persistent infection that we might have, and then having like a real pool that we have all information, not just for specific TCR sequences, but also like what this TCR actually recognize, and that's just on the T cell. And then like the B cell is a whole different level, right? Like we don't even have the technology to be able to do this now, as far as I'm aware of. But then like we could understand a lot more, right? Like whether like do we need, I don't know, like just for the S protein, like antibody against S protein for like SARS coronavirus too, or like do we need other, and what B cell phenotype it will become, or in the context of like dengue infection, right? Like where secondary infection with a different serotype would actually lead to like worse outcome due to antibody dependent enhancement. So like having the antigen or the epitope information on top of the TCR or BCR sequences would be amazing. And I think that's like another the next level that I'm like expectant to see.
what are the challenges in collecting samples if we want to retain this kind of microbiota as well as the host immune system? I can imagine that the possibility for cross-contamination would be rife. How do we do that?
Yeah, I think that's a very important question, because we had this thing going on with our study, the contamination issue, and that's why the spatial transcriptomics comes into picture, because there, if we just preserve the samples in FFP blocks, that will be very helpful, and very low contamination can happen during that process. That's what we saw in our study. The other thing is that because here we are talking about the human system, the human cells and the microbes collectively, like together. So we know that a random contamination will be everywhere, but something which is interacting with the human cells will be spatially localized. So I think that's where the spatial technology will be very nice. But yeah, at the same time, if we go move forward with this, so it will be very important to basically talk about this and generate a pipeline where the samples are taken in the most clean manner as much as possible in the clinics. And that's why the FFP samples are better in that case, because they are immediately fixed. Yeah.
Spatial technologies themselves, lack a lot of resolution, as Ash pointed out. And so I'm wondering, how do we kind of push that technology so that we're sequencing the host RNA, we're getting resolution of the cell types. We're able to see the TCR or BCR repertoire in the spatial blocks and also the microbiota. You know, who is anyone addressing that at the moment around the table? Ash, do you want to comment then?
No, that's on my wish list for Christmas this year, Chris. So yeah, I want TCR sequencing spatially, particularly for us with interests in peripheral tissue, T cells and cancer. But no, I don't know anyone that's actually working on that, so glad to hear about it.
So bringing the discussion back to the Human Cell Atlas Asia meeting, I guess my question is, given the importance of the immune system to healthy development, Achia, and Melanie, to diseases, even diseases not discussed in this session like cancer, but certainly to infectious disease, should this be one of the things that we focus on as a community moving forward, understanding how our diverse microbiota across different regions in Asia might be playing a role in shaping our immune systems and also might be playing a role in responses to pathogens which are endemic to our areas.
If I could comment, I think the endemicity, I can't even say that word, is important and is an opportunity, particularly if you talk about diseases like dengue or malaria or other parasitic infections, schistosomiasis, for example. I think that makes perfect sense to try and examine dynamic responses to those infections. Do tend to wonder how one would adapt the HCA philosophy to that goal. I mean, I understand that one can look at fetal development because one can sometimes access those tissues, but even with that's a well-controlled system, and you can hopefully get, well, usually a well-controlled system, but you can hopefully get the tissues you need to map that. But if the responses to pathogens are so varied, how can you possibly incorporate that philosophy into HCA? I can't see it. I don't know how to do that.
Arguably, we can't understand a healthy immune system if we don't understand what they're interacting with and how they're being trained. So perhaps this is a bit of a blind spot in the Immune Cell Atlas that we in Asia could actually push forward on.
Before that, I mean, I might have a comment on, I mean, microbiome does definitely contribute, but even before that, there's the genetics of the patient as well, right? Or the sample you're collecting. And this is quite well understood with these immune war atlases, that there is a significant component of genetics to how the immune cells are reacting. Their EQTLs spread out, right? So I think that would be the first stage to sort of incorporate, and then maybe some kind of an immune imputing framework, and then go on with the microbiome. Probably it's possible to capture.
Multimodality, including host genetics, inflammatory responses, and I'm going to keep pushing for dynamic systems where we're actually looking at what the cells respond to. We're at the end of our session. Thank you so much for hanging on and participating in what I found was just absolutely exciting. I'd particularly like to highlight the fabulous early career researchers we have around the table today. And thank you very much. I hope you join us shortly for the final wrap-up session with the chairs of the other speakers of the other sessions, talking about where to next for Human Cell Atlas. See you soon.
Thank you.